College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University Ludhiana has taken an initiative to prepare technique-based educative videos to cater the demands of undergraduate, postgraduate and doctorate students. These videos will help the students to understand concepts, principles and steps of various molecular techniques in a better way. Further, the videos are aimed to make the students self-sufficient and have them a feeling of hands-on training while they are away from the labs. To achieve this goal, Cell and Molecular Biology Lab of College of Animal Biotechnology has taken a step forward. This lab mainly focuses on the regulation of gene and protein expression through various advanced molecular biology tools. In this video, we will be discussing about enzyme-linked immunosorbent assay, which is commonly known as ELISA.
ELISA was developed in 1970s as radioimmunoassay. It is a quantitative tool which is widely used in the field of medicine and the industries as a measure of quality control. It is an analytical tool in biomedical field which is used for the detection and quantification of specific antigens as hormones and antibodies as bacteria or viruses. It is a biochemical assay that uses antibodies and an enzyme-mediated color change for the detection of either antigen in each sample. Basically, ELISA is of two types, direct and indirect ELISA. In the direct ELISA, primary antibody is labeled, while in the indirect ELISA, primary antibody that bind to the target molecule is not labeled, but the secondary antibody which is used against the primary antibody is labeled. The other two types of ELISA include sandwich and competitive ELISA. Whereas in sandwich ELISA, an antibody to a target protein is immobilized on the surface of microplate wells and incubated first with the target protein and then with another target protein specific antibody which is labeled with an enzyme. Compared to direct ELISA, the sandwich ELISA, which combines antibodies to two different epitopes on a target protein, has higher specificity. ELISA is typically performed in 96-well polystyrene plates which will passively bind antibodies and proteins. If we talk about the principle of ELISA, it works on the principle of antigen-antibody reaction. Specific antibodies bind the target antigen. It detects the presence and quantity of antigen binding. Therefore, depending upon the antigen-antibody combination, the assay may be called direct, indirect, sandwich or competitive ELISA. The concept of ELISA is that it uses coupling of antigens and antibodies. It relies on the specificity and affinity of antibodies for antigens, where specificity explains about the ability to discriminate among diverse proteins and affinity tells about the ability to tightly bind to molecules. The key step in ELISA assay is either direct detection of antigen by adhering or immobilizing the antigen, or indirect capturing the antigen specific capture antibodies respectively on the well surface. For sensitive and robust measurement, the antigen can be specifically selected from a sample of mixed antigen via capture antibody, sandwich between capture antibody and a detection antibody. If the antigen to be measured is smaller in size or has only one epitope for antibody binding, a competitive ELISA is used in which either the antigen is labeled that competes for the unlabeled antigen-antibody complex formation, or the antibody is labeled which competes for the bound antigen in the sample. Each of these modified techniques of ELISA can be used for a qualitative and quantitative purposes. In ELISA, various antigen-antibody combinations are used, including an enzyme-labeled antigen or antibody. The enzyme activity is measured by calorimetry, in which a substrate that changes the color when modified by the enzyme. Once the substrate is added, the absorption of light due to the product formed is measured and then converted to numeric values. If we talk about the requirements for ELISA, first of all we require antigen, ELISA plate, then stop solution, substrate solution and secondary antibody. The other materials which are required to perform ELISA are an incubator maintained at the temperature of 37 degrees Celsius, absorbent paper, precision pipettes and disposable pipette tips, clean tubes, deionized or distilled water, and then in the last but not the least one, microplate reader with the wavelength of 450 nanometers. Coming to the procedure of ELISA, to begin with, 50 nanogram of protein which act as an antigen is added in wells along with 100 microliter of sodium bicarbonate. Then the sample is incubated for overnight at the temperature of 37 degrees Celsius. The next morning, empty the plates and then blocking is done by adding 150 microliter blocking solution in each well. Then the plate is incubated for 1 hour at the temperature of 37 degrees Celsius. After 1 hour of incubation, the plate is washed with washing buffer. After washing, then sera is added and dilution buffer in each well. After the addition of dilution buffer, we re-incubate the plate for 1 hour at 37 degrees Celsius. After this step, again plate is washed with washing buffer and then 100 microliter of horseradish peroxidase conjugated secondary antibody is added at the dilution rate of 1:3000 in dilution buffer in each well. Followed by incubation again for 1 hour at 37 degrees Celsius. After the incubation of 1 hour, the plate is washed with the washing buffer. And then 50 microliter of TMB substrate is added to each well. After this step, the plate is incubated for 3 minutes at the temperature of 37 degrees Celsius. Then the reaction is stopped by adding 50 microliter of stop solution, that is sulfuric acid, in each well. And then addition of stop solution, the plate is read immediately for the absorbance at 450 nanometer wavelength. After taking absorbance, a standard curve and R-square value is constructed by plotting absorbance against concentration. The value of R-square is used to calculate the concentration of serum samples. If we talk about its applications, ELISA is a useful tool for determining concentrations of antibodies in serum. It is further used in toxicology for rapid presumptive screening of certain classes of drugs. ELISA is popularly used in food industry to detect the presence of allergens which are legally required for labeling of ingredients. ELISA is used for the detection of viruses. It was the first universal testing kit developed for the diagnosis of HIV. ELISA kits employ the detection of human serum cystatin C to identify patients who are positive. ELISA is used in the hormone assays. Major advantages of ELISA over the other techniques is that no radiation hazards are involved during ELISA. It is easy to perform. It is both a qualitative as well as quantitative test which can demonstrate the amount as well as the presence of target antigen in the sample. ELISA is highly specific and sensitive test. It can detect the concentration of target antigen even in picograms, that is, one can say at very low concentrations. Equipments for ELISA are widely available. Last but not least, it can be used for various biological samples such as urine, blood, plasma and serum, etc. talking about the limitations of ELISA, which are few for ELISA. It may give false results with mutated or altered antigens. Insufficient blocking of immobilized antigen leads to false results. Enzyme substrate reaction is for very short time. Therefore, plates must be read as soon as possible.
To watch more educational videos on veterinary and animal sciences, subscribe the channel and please touch the bell icon. Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana on YouTube.